Okay, so today we are going to study glycogenesis. Okay, so what happens in this is that we have glucose here. After all the breakdowns, for example, whatever carbohydrates you have eaten, they break down to the basic component that is glucose. For this lecture's purpose. Okay, so now this glucose follows different kind of fates. For example, it can be broken down and convert into pyruvate and then enter the Krebs cycle, PCA cycle, okay, and then release energy, okay. Another pathway is that we can convert pyruvate back into glucose, okay, that will be called gluconeogenesis. But what today we are going to do is we are going to make glycogen, okay. So this process is glycogenesis. What happens is that Uh, first of all, the glucose was meant to go down this drain, but because we have now excess of glucose and different kind of hormones, and our body signals this glucose to be converted into glucose. But what happens is that the glucose has already been converted into glucose 6-phosphate by hexokinase enzyme or glucokinase. Okay, so now this glucose 6 phosphate, we don't need it down this cycle to produce energy. We need to store this particular form of glucose. So what do we do with it is that we convert into a different kind of glucose. Okay, we just try to mutate it. For example, this glucose, the normal structure of glucose is somewhat like this. This is the sixth carbon. First, second, third, fourth, fifth, and then sixth carbon. And over here we have the oxygen. So basically, in glucose 6 phosphate, we have phosphate attached to this particular sixth carbon. But if this particular structure persists, it means that it will follow this pathway. But we are not trying to utilize glucose. We are trying to save glucose for our future. So for that, what happens is that this phosphate will be shifted on position number 1, okay, and this will be removed from here. So what happens is that glucose, the structure of glucose has kind of mutated. It has changed its shape. Now the glucose we call glucose 6 phosphate because at the number 6 phosphate was incorporated. But now if the phosphate is on position number 1, we will call it glucose 1 phosphate, okay. And the enzyme which is going to perform this particular function is okay, we can name it easily because we are involving three things here. We are involving phosphate, we are involving glucose, and we are changing the shape of this particular molecule. So what we are going to call this enzyme is phosphoglucomutase, okay. This is going to mutate or change the shape of this glucose 6 phosphate structure. So what are we going to call this thing? Phospho, because the phosphate group was taken from position number 6 to 1. Gluco, because this is a glucose molecule on which this particular thing is happening. And mutase, because this particular molecule mutated into glucose 1 phosphate. Okay, what happens with glucose 1 phosphate is that it will be then converted into this will be converted into one particular thing which is called UDP glucose. Okay, now we were on glucose 1 phosphate, but suddenly we converted into glucose 6 phosphate. So how did we jump from glucose 1 phosphate to UDP glucose? This is because we incorporated a molecule which was known as GTP. And what is the GTP? It's actually something like we are already familiar with ATP, and what the function of ATP is that it will release the phosphates when needed. It will release the phosphates to turn up the turn the machinery or to provide energy to our cellular machinery to work. In the same way, we have just like we have ATP. ATP is made up of adenosine triphosphate. We have GTP. GTP is made up of guanosine triphosphate. So when this particular molecule will release phosphates, it will also release energy, and that will power different processes of our cell. Okay, so what happens is that this particular glucose has already one phosphate, and GTP has the molecule of GTP has three phosphates. Okay, so what happens is that this GTP loses two of its phosphates, inorganic phosphates, and only GMP combines with glucose 1 phosphate. Now, when glucose 1 phosphate combines uh, with GMP, what results is that two phosphates become the diphosphate of this GMP, and glucose remains such. Okay, so in all the reactions in biochemistry, we will be seeing that UDP particularly acts as a glue. Okay, this is going to attach this particular glucose in a chain. Next up, we have a different kind of molecule that is known as glycogenin. Okay, this particular molecule is derived from tyrosine. Okay. Now, this glycogenin has one free end on which there is presence of hydroxyl group. And wherever this OH group is present, we know that this particular hydrogen has tendency to lose itself from this structure and make this O charged, where the glucose can or any other molecule can come and attach. So what happens is that this glycogen then reacts with or encounters UDP glucose, and what happens is that we as a result get a glucose attached to a glucose that becomes attached to glycogenin, and UDP leaves. Okay, so because at this point we have started to make a molecule of glycogen, we will call the enzyme which will catalyze this particular reaction will be glycogen synthase. Okay. Okay, so now we can just conveniently elongate this particular chain of glucose, and this is the linear elongation of this particular chain in one direction. And how is that so? Because if we move back to the structure of glucose, for example, this is the structure of glucose. This is carbon number 1, this is 2, 3, 4, this is fifth, and that is sixth. What happens is that we are making new bonds at position number 4. At position number 4, the first carbon of the particular um, glucose will come and attach. So this is the alpha 1,4 bonding. Alpha 1,4 bonding will only be linear. Okay, so this is here we have the linear branching. Okay, but what happens that if we want to branch this chain, because as we are all familiar that the glycogen is a branched molecule. So for the branching of this particular thing, we have an enzyme, and quite conveniently, we have its name is branching enzyme. Okay, what branching enzyme does is that this is the particular glycogen molecule, and glucose is attached on it. What the enzyme is going to do is that branching enzyme is going to attach a glucose molecule here and branch, and the glucose synthesis will then try to make this chain longer. Okay, how are we able to make this particular branch is because we have okay, so now we know that position number 1 is occupied and 4 is also occupied. We only have 2 and 3 and 6. So, position number 2 and 3 are not actively participating in this particular glucose molecule. So we only have left position number 6. What happens is that at position number 6 we have another molecule of glucose attached. So this particular bond over here is glucose 1,6 bonding, and by this kind of bonding we are able to branch this particular glycogen molecule. So this is how we are able to get the central portion of this molecule is glycogen, and we are able to get glucose chains all around. Okay. So this is how we are able to get a glycogen molecule.